Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm Peter Bannattini, and here I discuss all things brain imaging with scientists from around the world. Today, my guest is Nathan Spreng. Dr. Spreng is the James McGill Professor of Neurology and Neurosurgery and Director of the Laboratory of Brain and Cognition at the Montreal Neurological Institute and Hospital at McGill University. As an undergraduate, Nathan was initially interested in pursuing a major in poetry until he took a psychology class that sparked his interest in the brain. He received his Ph.D. in 2008 from the University of Toronto in Brian Levine's lab and pursued postdocs with Cheryl Grady at the University of Toronto and Dan Schacter at Harvard. After about 5 years as an assistant professor at Cornell University, he moved to McGill University, where he is now. Throughout his career, Dr. Spreng has been using fMRI to reveal subtle, yet repeatable large-scale brain networks as they relate to attention, memory, cognitive control, and social cognition. He's also helped to elucidate the central role that the default mode network plays in self-generated thought and how it dynamically interacts with multiple systems in the brain. Here we have a fun, far-reaching conversation about his work and what it implies, covering his study of age dependence of resting state hippocampal-linked network ensembles, how to move from mapping networks to modeling and understanding mechanisms, the many possible clinical implications of his work, current understanding of Alzheimer's disease, our mutual appreciation for multi-echo EPI, his data release paper of a large multi-echo EPI and structural MRI data set, and much more. So I hope you enjoy it. So welcome, Nathan.
Well, thank you, Peter. It's really great to be here.
So I usually start these podcasts with talking a little bit about people's history, and I think now you're a full professor. It seems like you've been making this progress. You've been making rapid progress with that, and you're well published and doing very well. But at the same time, you've had sort of you've had a few fellowships. You were at Cornell, and now you're here. So, but even before that, we were talking a little bit ahead of time, and it sounds like your early neuroscience sort of career was a little bit eclectic and pretty interesting. So what first got you? So let me start out by saying, what did you initially want to do with your life after maybe when you were in college, and how did you first get into brain imaging or start becoming interested in brain imaging?
Well, I think actually going pretty far back to high school, I was very interested in being a professor. Although at the time it was really for very different reasons, and I didn't really know what it meant. But I've always been involved in the arts, and I grew up in San Francisco in an art community where I was writing and performing poetry and working with noise musicians and doing some performance art. And I went to Sarah Lawrence College to continue and refine that craft, especially with writing. And I was studying philosophy at the time, and Sarah Lawrence has a very unique educational model. Everything is seminar based, and you only take 3 courses at a time. And the idea is to go deep. So I was taking a poetry class, philosophy, and I also happened to take a cognitive science class with Elizabeth Johnston. And I loved it. I just found it to be so fascinating, this link between how humans are and how they behave in the world and its relationship to the brain. And so I started digging deeper in my next course, took a neurobiology class, and I actually remember going to visit my friends one summer. This is after my second year. Everybody had moved up to Portland because San Francisco was no longer affordable. So that whole Portlandia, the spirit of the 90s is alive in Portland, is a very real thing.
Yeah.
And I remember almost coming out to my friends and saying, like, look, like I don't really think I'm an artist anymore. Like, I'm a scientist. And I was really nervous. And they were like, it's okay, Nathan. Like, we still love you. And I went back my third year and I took another class with Elizabeth Johnston. And this was on memory, human memory. And I remember reading a case study that was published by Endel Tolving. And it was about patient Casey, but in this paper, it was referred to as NN. And it was this accounting of an individual who had lost the ability to recollect any event from his past. But he also had this really interesting co-occurring deficit that he couldn't imagine the future. So it's written in this article that, Casey, he's asked, like, what does it feel like to be asked to think about the future? And he says, it's like being led into a room and told to sit down in a chair and there's no chair. And
wow.
I just thought this was so fascinating. It's like, why is it, or how is it that if you lose the ability to recollect the past, that you would have this inability to imagine the future? Huh. And it was this question that led me to do a senior thesis. And we didn't actually do research at Sarah Lawrence. So this was a literature review. Okay. And trying to put the pieces together with regards to memory function and interactions with the frontal lobes. And in retrospect, so when I was finished, I realized I wanted to go to graduate school. And I took a year off, did something that I don't recommend anyone does. Is like I just went to England and tended bar.
Wow. Well, it sounds like a great thing. I mean, it sounds like, you don't recommend it, but it's still at the same time, it probably was maybe it was useful.
It was wonderful. I mean, I was in Oxford. I was pulling pints at the Bear, which is one of the oldest pubs in the country. Yeah. But I was reading The Economist and the Herald Tribune and not doing much science. But I was applying to graduate schools. And what I realized was half of the people that I cited in my thesis were all at the University of Toronto. So this included like Endel Tolving, Morris Moscovich, Brian Levine, Randy McIntosh, all these folks that were really involved in human memory and also basics of network neuroscience. Yeah. And I was fortunate enough to be admitted with these ideas, these theoretical ideas, but absolutely no coding experience and a pretty shallow education in statistics. So I think that's where the liberal arts training is quite different than I think what a lot of people have. And, it's something that I grapple with a lot in terms of awareness of my strengths and weaknesses and what I can contribute to science.
Yeah. Yeah. So, I mean, that was, right. That was about 2003 when you started in Toronto, but that's the thing though. It's like, I actually think that, right. Recognize your weaknesses and trying to work with that in that sense. I mean, you can always learn coding and people have become great coders, after a couple of years, just, taking classes or whatever. But, that's it. Right. I mean, that's sort of an example of what, all of us kind of do. We're always on the edge of things we don't know in science. And we're always on the edge of, skills that we don't know. And you need a team to sort of work together. And it seems like to me, the most important skill, and this is what I tried to teach people as well. I mean, it seems like what you had at the very beginning, what you were developing is, communicating and thinking clearly about things. I think that is sort of underemphasized. I think when people go into graduate school to do science, they think, I can just, do experiments and figure things out. But there's so much writing and there's so much, iteration of thinking and thinking clearly and asking really good questions that I think your background probably helped you a lot for, as far as that's concerned.
It did, and it required a lot of reading and just trying to understand the space of inquiry. Like, what is it that we know about these spaces, and where are these like large knowledge gaps that we may not even be aware of? And
Yeah.
I think that's where I think a critical thing is to have good colleagues where they can share their knowledge as well, recommend some papers. There's going deep into reference lists, and I mean, I still love some like just the old classic papers. Like one I assign to my lab every other year or so is Marcel Meslim's From Sensation to Cognition.
Yes. Oh God, yes. Yeah, as a matter of fact, I have to admit, I actually didn't fully appreciate that paper until I had to have, I was interviewing Marcel on a podcast, and I took a look at that paper, and oh my God, it just blew my mind,
Yeah.
how clearly written it was, and how, oh my, it just was, yeah. So anyway, so I
mean, it's a beast too. Like I think the first time I read it, it took about 18 hours. Now that I've read it about 6 or 7 times, I think I've got it down to 4. It's a fun journey with my trainees to talk about these ideas, and I think so much was so well articulated. A lot of these older papers have that, and I really feel lucky that I was assigned them in my graduate training.
yeah,
We learned a lot about connectivity, looking at retrograde and anterograde tracers in macaques.
yes,
That
yes,
I think richly informed. I mean, Pat Goldman-Rakish had a map essentially of the default network long before we had fMRI estimations of it.
oh really? Oh, I didn't
Yeah.
know that. Huh.
I mean, it wasn't referred to that as such, but looking back, the pieces fit together and it makes sense.
Yeah,
So
yeah.
I think
Well,
it's a challenge to stay on top of the field with all the papers that are coming out now, but also digging back into the stacks and pulling out some of those real gems. I think is
yeah.
an important part of training.
Yeah, and it's not like it's interesting too, because you always think, oh, well, it was old and therefore it's not really that relevant. And certainly certain parts are always, not exactly up to date. But it's amazing how, in some ways, some aspects are more advanced because people had to sort of think that, they had more space to sort of construct these clear models. Whereas now I think the style of papers are, you make sure you do your statistics right, and you sort of say something, and then you say, well, it agrees with that, and it's, it might be this, and then that's it, and which is great. But there's, I think when you go further back, I think people maybe tried to, or the papers at least that lasted to be classics are the ones that sort of, gave insight.
Mm.
Uh, and I actually, that's what I always bug my students and postdocs about. Like when they write a paper, an abstract, it's like, okay, you should try to convey some insight, not just data and not
That's
just
right.
results, but insight. But
And I encourage them to take a risk. Try to be bold. Like, say something about what you think it means.
yeah,
It can be scary, and it's okay to be wrong. But these findings mean something. Like we're generating knowledge from our data, but these have implications for who we are as humans and how we live in the world. And I think it's important to reflect on what that is and what we found when we do this work.
yeah, I completely agree. So okay, so why don't we get into some of your, I mean, you've, just looking at your overall biography. I mean, it seems like I was trying to sort of put it into a category. I mean, obviously you're a cognitive neuroscientist. You were trained in Toronto. Now you're at Montreal Neurological Institute, and you do mostly fMRI on humans, and also structural MRI and things like that as well. And there's a great group in Toronto, and you do. It seems like you've been working on memory, default mode network, getting into mind wandering. Some recent papers on Alzheimer's, which I'd love to talk about as well. You have a big paper in which you released a large data set with multi-echo, and then finally you have other papers that can't get into on more social cognition and things like that, which we might, who knows? But maybe let me start with a paper that you pointed out to me, and I thought was just, I had a chance to look at it last night, and it was just amazing. Looking at, there's a lot to it too, and I really love the style in which you compare young and old, introspective, and sort of like different types of memory, and then looking at the ensembles and the connectivity that's associated with that. You didn't do the task during you looked at resting state, but then you looked at how well they did on performance either afterwards or before when you tested them. If I can encapsulate that. But why don't you talk about it? This is the PNAS paper, hippocampus and temporal pole functional connectivity is associated with age and individual differences in autobiographical memory. Came out in 2022. I'd recommend reading it because not only is it a lot of data, but I think it struck me as a really you're sort of constructing the resting state connectivity model with the performance in several different ways, and I thought that was just wonderful how you built this model.
Well, thank you. I mean, the work. I mean, I'm really proud of this paper. This is the methods that went into it were many, many years in development, and I'm really grateful to Roni Seaton and Leticia Mulambo-Chilobo, who were my graduate students at the time in my lab, who did a lot of this work. This is in close collaboration with Gary Turner for data collection. And this really comes from work I've been doing from the beginning, which is really trying to understand first that connection between memory and future thinking, which led me to my postdoc with Dan Schacter, looking at imagination and how memory is not just a process of overt re-experiencing, but it's really a reconstruction of the past. And the way that we imagine the future is it's a novel construction of these disparate elements and ideas that might be perceptual or narrative based. And I've been really fascinated with autobiographical memory as a way to understand one's story and who we are. And the way that people recount events, there are tools by which we can that are empirically validated to reliably assess what the features are in an autobiographical narrative that are indicative of that re-experiencing of the past versus elements of our memory where we reflect upon the things that we know.
Yeah.
That doesn't necessarily involve traveling back in time and re-experiencing it. So when we share our memories with other people, we're bringing in part a recollective experience and also scaffolding that narrative with knowledge and pieces of information to help bridge and bring people along through that process.
So just to have a question that occurred to me here is, I mean, isn't at some level, like all of our memory, doesn't it start as autobiographical memory and then sort of, because everything is our experience of the information, and then eventually it becomes sort of abstracted to things we just know. Is that, or can you, I mean, for instance, even if you learn any fact, I mean, sometimes I associate where I was when I learned that fact, or the act of learning that fact. I mean, it seems like there's a mixture in some sense, but maybe there's more of a differentiation. So.
Well, I mean, there was Endel Tolving recently passed away, and he was really a pioneer in identifying and dissociating episodic memory from semantic memory. And when I was a graduate student, I had just the wonderful opportunity to go and sit in his office and talk to him about memory and science on many occasions. And he would believe there's a fairly strict separation between those two systems, but I think we have an evolving understanding that there's a lot of integration and there's extraction of gists that can go on to form memories. And yes, there are things that we learn simply as veridical knowledge, and it can be tagged to the source of when we learned it. this is actually a really important issue nowadays, especially with what we think about we know and the veracity of information that we acquire, especially with things online.
Yeah.
But yes, like all of these pieces, this of recollective experience and the conveying of knowledge are part of the stories we tell when we talk about our life. And I've been really interested in how this changes across the lifespan, and the necessary role that the brain plays. And so the hippocampus is critical for that recollective experience, and the temporal poles have been implicated in aspects of narrative, and some would argue it's a semantic hub, particularly in the left hemisphere. And these temporal lobe structures are densely integrated with the extended default network. And you see the default network robustly engaged during recollection. I think that and potentially aspects of social and mental inference are the two kinds of tasks that really drive this system, although in interesting and dissociable ways. So I was interested in looking at, this is just a basic premise I think we have, is that part of why we vary as individuals is because of variability in our brain. That like your brain is not quite as functionally interconnected as mine, and that has consequences for how we go about and comport ourselves in the world.
Yeah.
So given that there are such interesting differences in how people tell these stories about their life, we wanted to then relate these measures to the functional connectivity of these key temporal lobe structures with that of the default network.
Okay.
And so we see really nice like dissociative patterns where these like recollective experiences are related to a certain ensemble across younger and older adults, whereas like the semantic components show a different pattern of connectivity in ways that seems to cohere actually well with activational studies. And then we also see a unique ensemble for memory in young people that seems to be quite distinct from when we talk to older adults and listen to their stories. So what this work really brought together methodologically was, I think, a lot of the challenges we have in the field and we've been battling with. And this is why, on the one hand, I turned to using multi-echo fMRI. When I was a postdoc with Dan Schacter, I stopped collecting data in 2011 because I really wanted to look at the functional architecture of the brain using resting state, and I really wanted to understand age-related differences and individual differences, and the whole issue of motion and global signal regression was just ruined everything. Basically, you could no longer really compare young and old because older adults move more, and global signal regression distorted the matrix and didn't really allow for you to make these kinds of group comparisons, let alone look at individual differences. So I just kind of hunkered down and waited for a couple of years to see what solutions emerged. And the 2012 neuroimage paper from your lab with Pronto Kundu really was like, the solution has emerged.
Well, it's a progression towards a solution, but
it felt a lot more tractable than just the multitude of pre-processing pipelines that were not as satisfying. I mean, we tried other approaches, a natty core and whatnot, but really, I think that change in acquisition has been key, because we know that, the temporal SNR of bold signal varies across cortex and by TE. So we can really optimize our signal quality by having multiple TEs and to get signal in places that are really exciting, like orbital frontal cortex and the temporal poles.
Yes.
And then the way that we can look at the TE dependence of bold and really separate the bold components from the non-bold components in the fMRI time series, I think is just absolutely critical, because there's a lot of noise in that signal. When I first saw the percent of signal that we're retaining, I was shocked. I mean, we're looking at like less than a quarter of the signal is really bold, and the rest of it just seems to be noise.
Yes. Yeah. And right. I mean, some people, you're right, there's pre-processing methods like pre-whitening and all kinds of other things, but you're right. At the end of the day, you just want to make sure, but even it's not perfect though, because, respiration might have a bold component, but it's still like global signal. So there's still work to be done. But
Sure.
yeah, I mean, I'm a big proponent of doing multi-echo whenever we can. And, we're trying to get the vendors to, GE is doing it. I think Siemens is, they're both doing it.
Yeah. Philips has a multi-echo sequence as well. I've actually been working with the Canadian dementia imaging platform, and we recently changed protocols, and we'll be scanning the Canadian longitudinal study of aging cohort, like a subset of them, with multi-echo imaging. So,
okay, good.
Yeah, no, a few thousand more people with multi-echo scanning, which is really exciting.
That is awesome.
But back to this paper. So
yeah.
one other methodological piece that I think is really important, and I think there's a growing awareness of this in the field. I'm not the only one who's thought of this, but this really emerged from an OHBM talk that Jim Haxby gave on hyperalignment. And that was really just the recognition that people's functional neuroanatomy varies.
Yes.
And I think if you want to capture individual differences in network architecture, you need to respect the fact that there's variability. And, this goes all the way back to Nancy Canwisher's first study looking at the fusiform face area. And those peaks vary across people in the ventral temporal cortex. Functional areas shift in their boundary, in their size, their delineation across people. And I believed that we could use resting state as a way to characterize functional neuroanatomy. And pairing up with Richard Leahy, we developed an approach which aims to maximize the homogeneity within parcels while respecting the network architecture across individuals, and critically preserving the labels of each parcel across people so that we can still continue to make a valid inference about differences in the network architecture.
Yeah.
So this approach that we call like GPIP, group parcellation. No, I always get this wrong. It's terrible. I think it's group parcellation with an individual prior. Is that it? Anyway, GPIP was
either way, right? Yeah, exactly. I'm trying to look forward, but but either way, group. Okay, let's see here. Either way, yeah.
I know.
They can find it.
We run in all these abbreviations and then having to reconstruct them sometimes.
I know. I hate, that's one of my pet peeves is in papers when there's so many abbreviations that you just lose track of what you're reading, and you always have to go back and check what the abbreviation is.
Exactly.
But one thing about I love that idea and I love that approach. As in fact, it's sort of tangentially sort of related to 2 years ago this paper came out by Merrick talking about the problem when you with it's called BWAS, sort of needing thousands of subjects, and it was a great paper, but it used a method of just having a standard template, a parcellation template, and warping all the brains to that template. And my first thought was, there's many potential other issues with, differences in populations and things like that with that paper. But paper was a great paper. But one thing that I thought they didn't quite emphasize enough maybe is that, there's a lot smeared out if you have 1,000 subjects and you're just warping them into a template and just expect all the parcels to just line up. And so what you did was sort of take this, template parcellation and sort of like, fit each individual parcellation as it was noisy because it was done in an individual, but as best they can with this parcellation template in some sense. Right. So it was, And I was just talking with the WashU group, and they do a slightly different approach. It's, they do deep sampling where they have over 5 hours of data. And so they have a different sort of parcellation method using, boundaries of resting state connectivity. Very similar though in many ways, but to other techniques, but it's their techniques. And they actually show in their data that individual parcellations are very, very, they're not very different, but they're different enough, as you show as well. It would be interesting to see if one could get away with, using your approach on, 2 sets of like 10 minutes or something, rather than needing 5 hours of averaging, and if you get the same results. But anyway, it would be, but they're both, I think, the right idea.
Yeah. Well, Chuck Lynch has done some really great work with multi-echo, showing how we get pretty good signal reliability with multi-echo a lot faster, with a lot less data. And yes, we absolutely needed, I think, some of these studies where people are scanned for 20 hours or 5 hours. And we certainly had to get past that point when we thought that 6 minutes was enough. But I think that we got a lot of gains with multi-echo. And in these studies, I was collecting 2 runs of 10 minutes. And we get good reliability there.
Okay.
We get our test retest is extremely high. Above like 0.9. So that's where I have a lot of faith in multi-echo as a way to get reliable estimations. And what I think is really important is that this really needs to be done in an amount of time that's clinically feasible.
Yep.
If we're going to be taking fMRI to the clinic, we're not going to be scanning people for 5 hours.
Right, exactly.
It's just not possible. So I think it's really important to, and I do a lot of work with aging and people in the pre-symptomatic stages of Alzheimer's disease. And I do think there is something meaningful that fMRI can contribute to our understanding of these kinds of populations. And it's a real challenge when, some of the benchmarking that's been done for like what's considered quality data is only really focused on young adults who are very compliant, have very healthy brains. And there's not as much of an inference that we can make from that when we're trying to understand special populations. Now, at the same time, I'm not trying to excuse bad data. There's plenty of data that's suboptimal, that maybe is, we've had effects obscured or potentially even identified spurious findings because the data quality was low.
Yeah.
So I'm a very strong advocate for just high quality data, but I think we need to always have the clinic in mind at a certain point, that this is a goal in the end. And I think, as I said, many hours of scanning is just going to be like too far beyond what's going to be clinically tractable.
Yeah. And so along those lines, I mean, I'm always convinced. I mean, I remember when Tom Insel was, director of NIMH, so every once in a while he would visit and be in my office, and he's like, fMRI is being funded, a lot. When is it going to finally be, useful clinically? And certainly it's in everyone, when they write their grants, they always put in a little blurb about how it's going to eventually be used clinically. But we're still, I mean, pre-surgical mapping a little bit is used, but there's it's such an important thing to start thinking about how either we can develop biomarkers for or ways of early diagnosis or something, maybe even it might be neurofeedback or whatever, to be used clinically, to put a person in a scanner and be able to say something unique clinically in a sort of a cost-effective way that you couldn't do otherwise. And I think we can. I think there's so much, everything from looking at basic physiology to advanced biomarkers of either schizophrenia or other disorders or Alzheimer's early indicators. Who knows? But it's just a matter of not only getting that, and as you mentioned, Prontic with multi-echo, it's like having multi-echo, but also having a platform that you can just use, that a clinician can just bring up and say, hey, this is the analysis, this is what we do. And yeah, it's so important. And then once that happens, I think what will happen is that all the vendors, like we were talking about, what vendors have multi-echo, they barely pay attention to fMRI, and because there's no clinical market. And so once there's a clinical market, then we will see wonderful changes in how, every sequence, every scanner will have vaso and high resolution 7 Tesla layers, whatever. But anyway, but I digress again. But let me just get back to this paper. So along those lines, So, so as far as what you found, okay, one other thought came to me as I was reading this paper and listening to you, is that uh, it, you know, so Alex Huth and Jack Gallant have sort of done different studies, very different studies, but in which they're sort of mapping out semantic space using single voxel predictive modeling with very detailed regressors with a task, with naturalistic task. But there, it's and it reminds me a little bit of Jeff Binder's work, in which he's looking at default mode network as it relates to personal, like, semantic space and everything. And I'm trying to sort of bring it all together into a model or at least a construct of some sort, as to like what's going on as far as like when you're encoding information. It's definitely some interaction of hippocampus with the rest of the cortex, either with the sensory experience or with the abstract semantic space. And then when you recall it, it's either, there's temporal poles as well. But I mean, so as far as, can you describe, I mean, it's maybe very complicated, too complicated to just describe in words, and what your general thought of is, when you encode information and when you retrieve it, and the main differences between young and old in this case. Maybe that's. I just want to try to get some sort of encapsulation of because it was really good, and maybe one just has to read the paper, because it's really it's you get a sense that there's some clear differences between young and old. And
there are, and if you listen to young people talk about their memories, it's filled with details. They're so detailed rich.
Yes.
And I might say that it can be tedious in some ways, how many details there are. Like I have teenage kids, and they'll be reading a book, and I'll say, like, "Oh, what's your book about?" And it's it's the whole play-by-play. And developmentally, I think we become better able to extract a gist and arrive upon that point and convey it more efficiently.
Yeah.
And as we age, we have this continuing accrual of knowledge and experience, and that information in healthy brains becomes like synthesized, and we can extract meaning and identify patterns that emerge over years and decades that we can then reflect upon and share as knowledge that doesn't necessarily emerge from one single experience. Yeah. So I think part of why kids and young people have such vivid memories is in part because they don't know what's going to be important later.
Yeah.
So encoding as much of it as possible is useful. And it's only over time, where there are these recurrent patterns, and these patterns begin to align, that I think you get that distillation of knowledge from experience.
Okay.
And as middle-aged and moving into older adulthood, we see that the patterns are really dominated more by our semantic structures and these things that we know.
Yeah.
And in some ways, we have a model of how the world works and are making predictions about the world, sometimes to the detriment of not actually experiencing it. We're experiencing the model in some ways, and don't necessarily have as much of that high fidelity perceptual information. So in a way, I think even as we age, we're not encoding some of those high resolution details as much, and in part because perhaps they're just not as relevant anymore.
Yeah. Yeah. That's really interesting. So, right, as we build this ever more sophisticated model of the world with more and more context and more experience, it's sort of like we have this structure, and we pay less attention to, our personal, the uniqueness of the personal experience in some sort of a linear way. That's interesting. And it probably, right, it ties very much into the differences in fluid intelligence and crystallized intelligence as it
Exactly. That's right.
as it varies. And also, just to jump to another, I think in your large data set paper, I thought, if I'm correct, there were some papers that suggested that looking at the the overall network structure between young and old. It seems that, and I don't know if this ties in at all, but young has more within network coherence and less across network coherence, whereas
right.
older people have less within network and more across network connectivity. What is that? What does that imply, do you think? Um,
Yeah, I mean, these have been observations going back a number of years now. Linda Geerlig and like Guggen Wig have both sort of put forward this idea of there's less segregation in older adult brains. So there's lower modularity of the systems and greater between network connectivity.
yeah.
And in some ways, I think some of these patterns are adaptive, and in other ways, I think they're compensatory for some aspects of declining brain health. So we have a study showing that the higher levels of white matter lesions, white matter hypertensities, is associated across like getting older with more between network connections, more between network functional connections. However, I think in some regards, some particular connections are helpful. So I think, and I have a theory that I've put forward with Gary Turner, which is the default executive coupling hypothesis of aging, where what we see is that there's more coupling between the default network and executive control regions in older adults. And we think that's because older adults are more likely to engage in goal-directed behavior and solve the problems that we're facing by drawing upon their knowledge. Whereas younger people they have a broader dynamic range. First of all, they're able to switch between systems much more readily and more flexibly, and can draw upon perceptual information and integrate that towards goal-directed processes a lot faster and easier. And they can also do the same with their knowledge stores, but again, they don't have the same depth and breadth of knowledge.
Yeah.
So
Yeah.
what we see as people get older is there is that denser connectivity between the default and executive control regions. And I think in part that's because of a repeated pattern of referring to what is known in our attempt to solve problems.
Okay. Interesting. All right. So, and it sort of brings me, to the next part of this is, and just in general, maybe it might be worth just sort of stepping back and talking about a little bit more of the default mode network.
Mm-hmm.
And you had many papers on this. You had a nice review article in the Annals of New York Academy of Sciences way back to 2014. And I know Randy Buckner and other people have been working on this as well. But just to focus on the default mode network, just in healthy people, I mean, it's not just one network. I mean, it seems that there's different subsystems: the core, the dorsal medial subsystem, the medial temporal subsystem. And it seems that they all have slightly different, it seems like the medial temporal is more involved with memory recall.
Mm-hmm.
The core is more self-referential, which is, and then the dorsal medial subsystem, like around the temporal poles or the hip, is more semantic, I guess, or comprehension or something like that. But so this is interesting in itself. And so what do you think, and this is a wide open question, one, and we were implying it by our talking about memory, about how the default mode network is sort of, it's tied with our subcortical systems, but it's connected to our cortex. It's sort of like our sense of self as we experience the world, to sort of encode things or to recall things from our personal experience in some way. But so is that what it does? I mean, what is the, I mean, if, what would you say is the evolutionary, the homeostatic purpose of default mode network? I mean, what is it actually? There's these subsystems, but what is actually, what mechanistically is it doing?
Yeah, I mean that's kind of the $1,000,000 question, and it's what are the functions of the default network are something that I've been pursuing now for 15 years or so. Actually, going even all the way back to my initial question as an undergraduate, which is like, what was the relationship between memory and future thinking? Well, it's they're both engaging the same network, which is the default network. And as a graduate student, actually, I remember when Randy Buckner's paper came out in Trends in Cognitive Sciences on self-projection, and he attributed these various features to the default network, which would be like stimulus independent thinking. So that could be recollecting the past. You're mentally traveling to another time, or imagining the future. You're not stimulus bound. You're out and imagining some other time, and also linking it to navigation. So the ability to simulate our movement in space, even though we're sitting still. And also, I think critically, he also stipulated that theory of mind reasoning, or the ability to transport ourselves into the mind of another person, is another form of self-projection.
Yeah.
And this was a really radical idea at the time when it came out. I was really excited about it because here was now a framework by which I could understand the link between memory and imagination, but also the social. And this was something that I hadn't really considered before that paper came out. And to the point where one of my very best friends in graduate school, Raymond Marr, was studying social cognition and narrative. And we'd known each other for years, played poker every week. And all of a sudden, it seemed like our research was very aligned. So in one of my first papers, Raymond Marr and Alice Kim and myself conducted a quantitative meta-analysis of the literature when the Ale approach just came out, and showed that there was convergence across these different task domains in the default network. And so a lot of the subsequent work has really been trying to understand, okay, like what is this system doing? And yes, like topographically, are these dissociable yet integrative functions? Yeah. And it does seem that yes, there may be this dorsal medial system that is involved in aspects of narrative, but also in social cognition and mental inference. And we have this other discrete subsystem that's really rooted in the medial temporal lobes to support mnemonic processing, but also includes imagination, which are potentially bound in these more midline hubs. Now that's something that's been disputed and it's being debated. The integration of semantic information as part of this system as well. Yeah. So there are, if we look at task-based activation, we can see dissociable functions related to language, related to social, and related to memory. But against the background of the rest of the brain, all of those functions are much more deeply integrated with each other than with the other systems. And I think that conveys something really important. And part of that is, I believe that the most important information for us as humans is the social. It's extraordinarily valuable for us to encode other people around us. Yes. And to rapidly form these representations of others in order to help how we navigate the social world. And these are very rapid changes that can occur in the contingencies, like who is aligned with who, and being able to link knowledge about someone that maybe you didn't see personally, but you heard about it from somebody else. Yeah. So all of this is really critical and necessary for us to effectively navigate the social world for our own success. So that's why I think even though these systems are dissociable at a certain level, I think it's their integration that gives rise to things that are really important to us and as humans and in our success. And something where we also derive a profound amount of meaning too.
Yeah. Yeah, it almost seems like it ties in. I mean, right. I mean, I've just been reading some books by Michael Graziano at Princeton. He's basically has these theories of consciousness, and one is very much tied in with our social, our need for our sense of others, and sort of projecting ourself, in having a sense of self, so that we can sort of model the senses of others well. But it seems that, without even bringing that point up at all, it seems that and he actually has some specific areas in mind, but certainly the theory of mind I always thought was more prefrontal, but I agree. I think that it's obviously very complicated. You can't point to any one spot, but I think that the default mode network, and also some people feel that it's more of a the thalamus is the one that sort of controls on some level. Cortical thalamus coupling controls this sort of integration of your personal experience with information in the cortex. But yeah, no, it does seem like the default mode network, in some sense, like you were saying, it integrates it integrates our experience, past and future, but it also allows us to appreciate having a theory of mind in some sense. Mm-hmm. It's I'm still trying to fully get my head around how that is controlled in some sense. How what, like, for instance, when information comes in, I guess this is the $1 million question with modeling in general. Like, how does our brain know what parts to become engaged to, take in the information and make it meaningful, mm-hmm. And to encode it in the appropriate way, and then to use it. It's obviously, these are, and maybe this touches upon a question that I always bring up, with any of us doing brain mapping. We can point to these areas, and we can create some sort of a taxonomy of like tasks and functions as they correspond to areas predominantly, and we could point to networks that are involved, but I'm still wondering, people, your hardcore neuroscientists might say, well, all you're doing is sort of cartography. It's like, okay, that's great. There's this area, there's these sub areas that are associated with these tasks, and it might be useful clinically to go in and maybe make sure you don't touch that area if you're taking out a tumor or something like that. But what does that tell us? How close are we to use this information to tell us about mechanisms, like mechanisms of what's actually going on? What's the computation that's going on? Obviously, this won't give us the answer, but how might it complement someday when we do know the answer? How will this have been useful? I guess it tells us where to look. And there's more we can do with this information, but I'm just, it's sort of an unfair question because it's totally not answerable.
Yeah.
It's something I struggle with all the time.
Yeah. We all like the idea of having a mechanistic understanding. And I think, we have a lot of limitations from every side, right? So I think with bold signal being how slow it is, there's a beautiful amount of structure in bold that I think is conveying something that's meaningful. And I think there's a lot of dimensions to the structure of bold that can tell us a lot about how the brain is organized, as well as when we're driving it with tasks, these discrete functional topographies and specializations that is extremely important. So it can tell us, the entire literature preceding neuroimaging, we did lesion symptom mapping.
Yep.
We learned a lot about localization of function. And yes. I think with fMRI, we're able to see much more so how not just these key regions that are necessary, but the regions that are involved. And I think we're getting closer to seeing how they communicate with each other.
Yes.
And this, has the potential, I think, to open up another avenue of potentially like targets in the context of psychiatric illness. I think if we're going to TMS is a tool that we have to potentially shift some of these network dynamics. If we characterize that topography well, and we could identify where there's differential weightings between regions that might be more pathological, that we can shift those weights. And I think that gets us to where, yes, we have these beautiful brain maps, but what are they good for? Well, this might be something that they're good for.
Yeah, I mean, they're totally, yeah. I mean, clinically, I think you're right. I think you hit upon a really good point that it's very low-hanging fruit in some sense to say, okay, like with ADHD, for instance, if you have ADHD, your default mode network might be, you might not have control over how your mind wanders. And you could target, if you did some sort of, you could look at the either metabolism or use some sort of deep TMS of some sort or something to get at the default mode network in some regard or to regulate it in some way. I totally agree with that. And there's a lot of good work being done with that as far as that's concerned. And actually that also gives insight into, the functional relevant. The more we try to apply it clinically, the more we modulate it, the more we can, totally agree with that.
I guess I'd like to see is just a deeper integration, I think, across modalities. I think that's going to be really important. So I've been talking with Yosef Parvizi out of Stanford for over 10 years, and we've just now started collaborating in more depth. And he's implanting like electrodes. He's doing intracortical EEG. And when you mentioned the thalamus, like there's a couple leads in the thalamus, anterior thalamus, the pulvinar, in addition to the hippocampus and orbital frontal cortex and posterior cingulate. And we're able to look at the dynamics at the speed of neurons now.
Yes.
In addition to perturbating each node and seeing how signal propagates and potentially the directionality of these effects.
Yeah.
So that's a really exciting space that we can hopefully now, I think, potentially test some of these things using other modalities like MEG. I've just started working with Sylvain Ballier here at McGill, doing MEG and trying to do some data fusion across these different types. So with 7T imaging to have like fine spatial resolution with bold, and then align that with MEG and even potentially this intracortical EEG signals to get a richer understanding as to what the dynamics are that are occurring across like multiple scales, physiologically and spatially. I think we'll maybe get closer.
Well, you bring out a really good point. I mean, it's not an either or. It's not like, oh, fMRI, what is it? And it's really this integration and also sort of like this iterative process where you ask some really good questions and you learn a little bit more, and then you might modulate something else either behaviorally or in the measurement or in how you look at it and integrate a little bit, and then you learn a little bit more. And then it really is this process. And this is what I try to tell people in terms of when I try to argue. And I mean, I'm very biased, that how useful fMRI is and how useful this is as far as this is concerned. And there are people even doing, very ambitious modeling of the fMRI signal and connectivity of, Danny Bassett and Michael Breakspear.
Great work. Absolutely.
Yeah. And Randy McIntosh for that matter too. I mean, it's really, and so on those levels, the models, you feel like you're getting closer to, some mechanistic process in some sense. And then you have these big models like, with Friston, like you were inferring, like, the whole idea that the brain creates a model of the world and sort of wants to minimize surprise. And that's sort of like a more principled construct in which everything kind of makes sense in this. And it's interesting. You mentioned the thalamus and you're looking at feed forward and feedback. Certainly, I'm vested in doing layer fMRI. Mm-hmm. And I think, I would love to do more work with the default mode network in terms of, from the layers you can infer, potentially from the location, what's feed forward and feedback. So that is a whole other slew of data that might complement these models in that regard.
Yeah. Absolutely, yeah. Sam Audrain, who is she's a
Yes.
mage working with Alex Martin, is doing some really beautiful layered work um, in the context of memory and like hippocampal cortical dynamics that I think is really elegantly going to sort of expanding our understanding of these dynamics and give us just a richer portrait of what's happening, not just across regions, but across these layers of cortex.
Yeah. Yeah. So that's hopeful. And, obviously we'd like to do layers with multi-echo and super high resolution. And
Yeah.
I think we can get there. Let me just keep on going here a little bit. I sort of hinted at mind wandering a little bit, and I was really intrigued. It seems like you've worked a little bit with Kalina Christoph and Kieran Fox, and I believe, and I got it wrong, and I was thinking of it before. Back in 2015 or 16, when the meeting was, I don't know. Some time during that period, that was the best paper award
Right.
at human brain mapping. It was the Wandering Brain: Meta-analysis of functional neuroimaging studies of mind wandering and related spontaneous thought processes. So you spent a lot of time thinking about mind wandering, and once again, this sort of ties in with the default mode network. But as you suggest, though, there's a lot more than just that. It's not just the default mode network. It's there's and also I feel this taps into also a really deep question about what resting state activity is. So you might as well just I couldn't summarize the papers as well as you can. So you might as well, it's just a period of time. You had a paper in Nature Reviews Neuroscience in 2016, and also the NeuroImage one, basically saying that that the default mode network contributes to the stream of consciousness, but on the other hand, it taps into other areas that become sort of spontaneously active and then feed into other areas and as well.
Yeah. So I was kind of pulled into this space of mind wandering. It's because of the work I did, really attributing specific attributes of thought to the functions of the default network. So looking at imagination and recollection, and I think these are key processes that the default network engages in, and these are also some of the core contents of where one's mind goes when it wanders. So I think of mind wandering as more of that the movement of thoughts in a way. I think in its coarsest description, I think I did just mind wandering is another way of saying thinking, which can quickly become fraught down the path towards consciousness. And so I think we're really trying to steer clear in some ways of that, but really to understand like the movement of thought and what are some of the constraints that are placed on thought with regards to being goal directed versus having very loose constraints. And importantly, I think also some of the interactions that we have with the environment at the time that we might be thinking. So there are plenty of occasions where just letting our mind wander, I think, is an extremely important thing for us to do as people. And in some ways, we do so less because we're so stimulus bound in a lot of ways. And I mean, this is all the way back to the discovery of the default network. When people are attending to visuospatial information in the environment, the default network is suppressed. And we do this all the time with alerts on our phones and across our devices. But I think there is an important function to mind wandering in that our thoughts will be pulled towards or drawn to some potentially like unresolved issues. Maybe you had an interaction with a coworker the other day that it just didn't sit right, and your brain will work on that and just try to. It'll just look at it from different angles and maybe find a place where there's insight and resolution as to that nagging thought. I think there
That
are
is,
reasons why our thoughts are pulled certain ways. And I think it's important to give ourselves time to let that happen.
yeah. Yeah. I love that thought. I love that thought because it reminds me a lot of, and I think that, one, I totally agree with you that mind wandering is something that it feels something really healthy to do, that, it's somewhere between, you're not dreaming and you're not, focused on the outside world or even internal goals. You're just sort of letting your mind drift. And what I find fascinating about that is that there's a paper that Maurizio Corbetta, I think, was on, that he wrote a few years ago on, what's the purpose of spontaneous fluctuations.
Hmm.
And it was a really nice paper in the sense that, it was sort of trying to talk about, resting state fluctuations in general, in the entire brain, might just be ways of, the networks to tune themselves in some regard. There's sort of like, you have you're optimized, it's just sort of spontaneous firing through the network to sort of optimize it. But in some sense, mind wandering, when I was thinking about your papers, I had the thought that, maybe our mind wandering is, our glimpse, our conscious awareness of that sort of stochastic process that's occurring anyway, everywhere, left and right motor cortex. It's not like you're mind wandering about moving your fingers. It's just somehow, visual cortex, all these areas that were active are somehow naturally, spontaneously fluctuating and interacting. But the ones that sort of tap into our awareness, our conscious awareness, those are our random thoughts that occur. And so we're sort of perceiving our own resting state fluctuations in some sense. You know what I'm saying? It's like somehow our thoughts are our random thoughts that occur just spontaneously are, that's going on everywhere in the brain. But, our conscious thoughts of that's sort of a glimpse into this stochastic process that's going on.
I think that's part of it. But I think actually, this might be a less popular view, but I actually think it's a fairly small part of it in some ways.
Okay.
If you look at task, right, you're looking at a very small percent signal change that is sort of riding with these like low frequency oscillations across regions of the brain. And so while, yes, what we think about or what task we're engaged in, and if you look at those oscillations, you have some ability to predict where those signals are going. But I also think that resting state fMRI is carrying information about like polysynaptic connectivity, and that more of the structure of the biology, and there in a certain way how I think resting state can be used to explore like what is that dynamic repertoire. So I think there are dynamics that are occurring during rest that are extraordinarily meaningful, and
Yes.
the variability across cortex, I think conveys very interesting things with regards to its function that may be related to what one is thinking or that wandering brain, but also is I think there's something deeper happening too. That's not just our thoughts.
Yeah. No, I totally agree. And I don't think our thoughts are necessarily right. I completely agree with that. I think that there's something, and that's actually what I was hinting with Maurizio's paper, that it seems like there's, like you were saying, it's something deeper going on that seems related. Like, for instance, there was that classic paper where they put people's arms in casts, and then they looked at the spontaneous activity across left and right motor cortex and showed that it went down. And it seems that the resting state fluctuations kind of are like an echo of like what's been going on that previous day or the, in terms of the network. And if there's less activity, then there's less connectivity. Who knows? And who knows what the network is doing to itself. I completely agree with that. And our conscious thoughts might be, and that was my thought, they might be something completely different, or they might simply be like you were saying, like if there was an unresolved conflict or some sort of thought, it might be the network working itself out in some sense. And we're just kind of aware of that. So it's like we're kind of riding on our thoughts as opposed to controlling our thoughts. I mean, as far as, when we mind wander.
Right. That's right.
Yeah. And there's a person in my group, Javier Gonzalez Casillo, who's sort of trying to, I think a lot of resting state is something more intrinsic, but also he's trying to tap in to moment changes in, thought content, which is a lot harder. It's really hard to even measure that during the scan, but, it's probably a fraction of the signal, but yeah, interesting. Yeah.
There's a lot of work that's done, like Johnny Smallwood and actually Jess Andrews Hanna doing those thought probes and really trying to see like, okay, what is it? I mean, we can certainly account for some of the variance at rest based upon the thoughts that people are having. And I think that's fascinating too, where, we have tasks that we've designed, as cognitive scientists or cognitive neuroscientists. And I think we tend to think about task states as these potentially like just discrete things. Yeah. But, in a lot of ways, I think our current set of tasks are somewhat limited in that what we're trying to do is just have a radically reduced version of how we do a computation. Whereas, living is a task in some ways, and it's filled with all that complexity. And that's what people come into the scanner with, is this history of activities. And that, I think, shapes what some of this underlying functional architecture is. And we can push it around the edges a little bit with tasks. Maurizio has done this work looking at specific visual field connectivity changes with visual training. If you show people lots and lots of faces or houses, you can get slightly different patterns of connectivity between those ventral temporal regions with prefrontal cortex. This is work that Dale Stevens did with Randy Buckner and Dan Schacter. So you can move these resting state correlations around a little bit based upon task. Yeah. But I think that's just suggestive of this sculpting that occurs in the functional architecture through repeated experiences.
Yeah. Yes.
So I think that's why it's no surprise that, large scale patterns of activation look like these networks as well.
Yes. Yes. Yeah. It's, and so I think the story is becoming clearer. But at the same time, I also think, there's just enough mystery as far as, what's really going. I mean, I think it might, the whole resting state thing might, once we figure out what and why it does this, I think it could, we could tap into sort of like the basic, the fundamental network properties of the brain as far as that's concerned. So I think there's much more here to explore as far as that's concerned, from not only looking for biomarkers, but also just understanding how the brain speaks to itself. I mean, why does it do this, and what modulates it? And it's so. Okay, all right. So why don't and also talking a little bit about clinical work, and I think it's really important. Alzheimer's disease. You've been doing some work with Alzheimer's patients, and you have a paper in 2016 in Nature Communications saying basal forebrain degeneration precedes and predicts the cortical spread of Alzheimer's pathology. And I know there's been a lot of papers looking at Alzheimer's pathology and how it spreads, and whether it's tau accumulation or is there clearance issues. How does this paper fit into what we understand today about Alzheimer's disease?
That's a great question. So I think a lot of my work kind of falls into two camps in some way, and they're interrelated, but something that has just grown with regards to what I see as important for us as humans is to understand Alzheimer's disease and to stop it essentially, because this is going to be impacting just a huge proportion of the population and absorb billions of dollars of our healthcare. And not to mention there's just the inextricable toll that it places on people and our caregivers and loved ones. So in a way, I don't know if I'm being naive and thinking that I have something to contribute to our understanding of Alzheimer's disease progression, coming at it with my background and experience. But the default network is certainly involved and a key hotspot for the progression of that disease. And this isn't my idea. I mean, this goes back quite some time. Like Randy Buckner has done some just phenomenal observations of this. And there's continuing work. I mean, a beautiful paper just came out in Neuron, I think a couple days ago, from Michael Breakspear and Bill Jaggist, looking at activation of hippocampus and the default network and discrete roles for amyloid and tau. Now,
let's take a look at that. Yeah,
there's amyloid and tau have gotten a lot of the press on this. And in a way, I think it's because we could image amyloid in living humans. And so rightly, it got a lot of attention. So I think there's been, an abundance of bias towards the amyloid hypothesis as a causal impact in the disease. And I'm not committed to that model. I think that it's important to understand a lot of other features and factors related to the disease. And I'm most interested in studying the pre-symptomatic stages. So those points in time where there's changes that are occurring in the brain before outright memory impairment. Yeah. And I think it's absolutely critical to start focusing on that window, because that's where we want to intervene. Because if we're going to stop this disorder, we want to do so before the damage is done. And this paper with Taylor Schmitz that you mentioned, we use structural data from the Alzheimer's Disease Neuroimaging Initiative, and there's beautiful longitudinal data in that. And I think longitudinal data is really going to be key to understanding the progression. And I really do think there is a role for MRI here as well. So there's a popular notion that's widely believed that Alzheimer's disease starts in entorhinal cortex in humans and then spreads from there. But what we show in this paper is that actually the basal forebrain is predictive of subsequent changes to entorhinal cortex. So if we go back and actually look at some of the BRAC staging, what we see is that there are earlier points of pathological accumulation. And we see this in structures like the locus coeruleus, as well as the basal forebrain. And I think these are really interesting and sort of almost underappreciated structures, because we know that, the locus coeruleus is providing the noradrenergic, norepinephrine modulation of cortex, whereas the basal forebrain is projecting the acetylcholine across the brain. And these sort of neurotransmitters and the neurotransmitter projection systems are often things we don't really think about a lot in the context of MRI. Like dopamine's gotten a lot more attention, but what evidence has emerged is that, Alzheimer's disease, it hits the default network, it spreads, and it's likely that these pathologies like amyloid and tau they're spreading transsynaptically. And the locus coeruleus is a structure that starts to accumulate tau as early as one in their 20s.
Really? Oh, wow. Okay.
And so we don't really know if it's both sensitive and specific and predictive of downstream decline, because everybody has tau in the locus coeruleus. And what I've been trying to do in my last number of years here at McGill is really trying to understand what is this pathological cascade that is occurring in these earliest stages of disease. So I've done a lot more work now with structural MRI in particular, some innovations with quantitative MRI, like magnetic transfer saturation,
Yep.
that is really amazing in terms of being able to characterize some of the white matter microstructure. And we can do so of these really small nuclei. I mean, the locus coeruleus is tiny.
Yeah, it's tiny. It's tiny, right.
And basal forebrain is bigger, but it's not a lot bigger. So these are structures that are hard to identify with standard MP RAGE, like T1 imaging. So
Yeah.
we need a lot of different methods for identifying these structures. So neuromelanin scans are helpful to get that contrast in the locus coeruleus, as well as the substantia nigra. But having good atlases based on histological work is really quite critical. Fortunately, these structures are well preserved across humans. There's not as much variability in terms of localizing them. Not like cortex. Cortex is tremendously variable, whereas these brainstem structures are at least they're more preserved across people, and really trying to characterize the integrity of these nuclei, I think, are going to be extremely important, as well as understanding their projection and really the fidelity of these neurons is so critical for healthy brain function.
Yeah. So do you think that, like, for instance, right, if you did some sort of a meganization transfer contrast with, or some way of looking at white matter integrity, gray matter integrity, somebody could go in potentially as a screening scan and get an image of the brain and actually say, okay, this is you have some potential early degradation in these specific areas using just MRI. And then if they do see that, I mean, this is what I'm always struck with. If they do see that, certainly there are therapies and drugs and things like that. But what can what can we do yet as far as about that? It's hard to know.
Yeah. Well, I mean, the goal really is to, I don't do this work, but what I hope emerges from the work is we can identify new intervention targets.
Yeah.
So the current drug that was approved recently for treatment of Alzheimer's disease, I mean, this is the first drug for treatment. I can never say it right. It's aducanumab, I think. And it clears amyloid beta from the brain.
Oh, really? Wow.
It does. It slows the rate of decline, but people are still declining. And a terrible side effect is brain bleeding.
Ooh.
And it doesn't seem to really work in women. So there's a lot of problems with this drug. And I think the main one is that it's the target is too far downstream. And I think we really need to figure out how to potentially promote brain health and the flexible fidelity of these areas that I think are hit first and hit hardest are these very large projection neurons in the brain.
Yeah.
If you look at a basal forebrain neuron and you took its axonal arborizations and you put them end to end, this neuron would be 100 meters long. I mean, it's just huge, and the metabolic demand on it is extremely high. Maintaining the structural fidelity of these neurons is a challenge, and with regard to lipidization, and they are also quite responsive to subtle rapid changes related to potentially attention, as well as subsequent memory formation and memory retrieval. So another my goal is to not just be able to use MRI to identify these structures and potentially characterize their health, but to have a complementary set of cognitive tasks that may give us insight into impairment that occurs prior to memory. Okay. So I think that attentional processes are actually going to be impaired before we see memory impairment. Okay. In a way, because you need to you need to attend to something before you encode it and then
Yep.
subsequently remember it. And we know that, acetylcholine as well as norepinephrine, these are necessary modulators for how we attend to the environment and encode information. So I think
That's interesting. Yeah. So you think it's actually could be like back to attention, like it could be, I mean, obviously everyone has different baselines of
Right.
there's people with ADHD, the people who are very good at encoding, but instead of, sort of, there's an indication that, maybe they're drifting off in how present they are in to attend to a situation, or they have some sort of low level task that gets at that. Either that, or I could even imagine even lower level, even some sort of a continuous attention task or
Mhm.
something, reaction time or whatever that could be sensitive. But yeah, I agree. How do you? But yeah, you'd have to calibrate it in some sense with each person, I imagine. Um,
Right. And that's,
but
I mean, one of the things that I find so amazing about working with older adults is that there is so much variability. And something I really love about that variability is it feels to me like older adults are very much themselves, and they're very different from each other in a way in a way that young people are in some ways like very similar.
yeah,
And there's some, it's almost like as we age we become more of who we are, and I think you can really see that when you talk to some older adults. But
yeah,
they're in clinic or in the lab, like their behavior is there's a lot of between subject variance. So a challenge is you might have somebody who is extremely smart, like very self-aware, realize that their memory is not what it was, and they could go to a neurologist and say, like, I'm not remembering well, and they're given a standard test, and they're in the normal range,
right, right,
and so they'll be sent home. And
yeah.
so
Even though they're alarmed, they're aware of the fact that they're not, and yeah, yeah,
so characterizing that between subject variance is going to be a real challenge, I think, and that confounds a lot of our biomarker developments too.
yep,
So I think we're going to need more than one modality. I think
yeah.
like a cross section of cognition, MRI, and potentially other biomarkers, I think, is going to be key.
Yeah. And I also think, I mean, as far as differentially diagnosing, I mean, can you tell the difference between Alzheimer's versus, progressive aphasia or
Oh yeah.
vascular dementia or, that sort of thing as well? Is there a way of either through imaging or behavioral, some combination to tell the difference?
Absolutely. I mean, these are very real challenges. I mean, when it comes to people presenting in the clinic, really trying to dissociate frontal temporal lobar degeneration from Alzheimer's type at the early stages it's a real challenge.
Yeah. Yeah.
Yeah.
And it
We
could have
don't have
different
clean
mechanisms.
subjects. The clinic's
Yeah.
messy.
Right, right, and but so just one last thought on that, and I've been thinking a lot about uh, you know trying to image, all probably all the fMRI physicists I know are, in the back of their head, one, you're trying to get direct neural current imaging or direct imaging, but the other thing is sort of now trying to get to look at the glymphatic system,
Mm.
and I also suspect that some people have ideas that Alzheimer's may not necessarily be a rapid accumulation of tau proteins or whatever, but more a inefficiency of the lymphatic system to clear them.
Mhm.
As we all produce them, we have good lymphatic systems that clears them out efficiently. Whereas with Alzheimer's, there might be problems with clearing it, and certainly there's an indication with lack of sleep where you don't get your lymphatic system active. There's
Mhm.
increased accumulation of tau tau proteins. Do you have any thoughts on that? I mean, it seems like even the location where it shows up first might be like an area where, like, CSF flows last, and who knows? It could be a bottleneck in where the lymphatic system might break down, or in some
Yeah.
sense. I don't know if you've had any thoughts on that sort of thing.
Yeah, I mean, I think the astroglial system is one that we haven't thought a lot about, at least, from a human perspective. There's a lot of non-human animal work that has looked at this, but I think aspects of like astroglial reactivity to the presence of these pathologies, like are they effective at like grabbing and chewing them up, or are they becoming inflamed, and is that inflammation can then cause more downstream disruption.
Okay.
And combined with aspects of the blood-brain barrier, like having reduced integrity there in the context of aging, I think there's a lot of multi-factor issues that come to play with brain health as we get older.
Yeah.
I am very optimistic, though. I think that we will figure these things out and look to like how we can actually promote brain health. If we look at the lifespan of humans, we've added 30 years to our lifespan in the last 100.
Yeah.
It's just remarkable how much older we are living. And this is through a lot of advances in the biomedical sciences. And I think if we can push through and solve the dementias, and I believe we will, then that opens up another number of decades where humans can live, and hopefully with a quality of life that makes that worthwhile.
Yeah, I completely agree. And yeah, certainly we are, we're it's ratcheting forward. Our imaging is getting better. Even with MRI, we're still finding better ways of pulling out information from the signal. And and so much of your work is sort of like along that forefront and right at the edge of making ever more refined models of cognition, of pathology. The group in general in Toronto that you work with is just awesome. And
well, I do have to correct you there. I am in Montreal.
I'm sorry, Montreal, that you used to work with, and also Montreal. Montreal is also awesome. I know.
Yeah.
I know a whole bunch of people at Montreal that are. I mean, that's sort of. I mean, they're both great groups, but I think actually Montreal might even be more established as far as the neurologic institutes and everything like that. So
it's wonderful. I mean, it's such a great place to do science because there's so many people with such a breadth of expertise that it's so much fun. I mean, this is what I love about the neurosciences. I mean, it's a lot of fun, and I love this space because now I'm working with physicists and I'm trying to like develop some new sequences to have even smaller voxels. I mean, we're trying to aim for like 0.8 millimeter isotropic, and to look at some of these dynamics, but also things that I never would have done before, like PET imaging. I'm not only leveraging work with Sylvia Villeneuve to look at amyloid and tau, but also a radio tracer that's FeOBV, which binds to presynaptic cholinergic vesicles. So for the first time, we can get whole brain measures of cholinergic tone, which is just amazing. And I never would have done this work before. I mean, I was an assistant professor at Cornell University and learned a lot there, had a wonderful time. And actually, there's an avenue of my research that I began pursuing there, looking at the vulnerability to financial exploitation in older adults. And that was really because of work with Carl Pillemer and his work in elder abuse. And so trying to understand aspects of neurocognitive aging as to what could render older adults vulnerable. So that was a really wonderful line of research that I continued to pursue. But shifting back into an environment that has a medical school and just all the wonderful tools for brain imaging, including MEG and PET, and expertise around biomarkers. That's just fundamentally going to change the field of aging are these plasma-based biomarkers now, where with a simple blood draw, you can now get measures of amyloid and tau, which seems to be sensitive and specific to what's circulating in the brain that we could only previously see with PET or with a lumbar puncture and analyzing the cerebrospinal fluid.
Oh, that's interesting.
The ways that we can characterize health and brain health is so incredible. And having the kind of colleagues that are here in Montreal is just, I couldn't ask for anything more. It's wonderful.
Yeah. So, and also just to reemphasize your point, I mean, with every new method, like even going to high resolution with MRI or with getting, having a new sort of piece of information, it opens up so much. And so then we spend the next 5 years catching up with relating these other techniques and trying to make a model of that. And so it's great. It's just the technology keeps on going. Our questions get better. Is there, just out of curiosity, is there any, you've worked both in the U.S. and in Canada now. How do they compare in terms of, the culture or doing your research or grants or things like that?
My wife is Canadian, and so my kids are Canadian. We were interested in coming back. It took some time to do so. I just, on a general level, having socialized medicine just makes the entire culture less stressed. And there's a little bit more care for the citizens and makes a palatable difference. But with regards to the granting agencies, there's similar parallel agencies. We have NSERC is like NSF, and then there's Canadian Institute of Health Research, which is like the NIH. Budgets are much, much smaller here. I think they're spending something around the range of like 2.5% of GDP on basic research, where it's 5% or more in the US, and that's a much larger GDP. So there's a lot more money in the US, and the grants are bigger. But the probability of getting a grant in Canada is higher. So it's a nice system. I think it's a reasonable system. But it's like, I mean, you have a different thing that you're doing within.
I'm lucky. Right. We just have a budget. I'm an intramural program at least, but
But what's funny is that when applying for grants, it just feels like the lottery in a way. Like when I started as an assistant professor, I applied for something on the order of like 32 grants before I finally got one.
Oh God.
And it was excruciating. It's not like those were all separate ideas. And in fact, over time now, nearly every proposal has been funded in some form or another. But I am fortunate in that I can actually apply for project grants both in Canada and in the United States, because the work that I do benefits Americans. So I've had complete crossover interactions with my grant success. I've had a grant triaged twice at NIMH and then funded on the first round at CIHR. And then I've had a grant that was go to CIHR that was ranked, like it was 86th out of 87, as in the second worst grant on the panel. It was held in such low esteem that I couldn't even get that upset about it. They just hated it. And then the same grant went to NIA and was funded on the first round. So
it just seems right. There's a little bit of noise in the system there. It almost seems like a perfect application for ChatGPT or something like that, to some sort of AI grant review system that's free of bias, or knows kind of everything. All the groups in Canada, everything from Montreal to Toronto to British Columbia, it's all I really respect. I mean, you're making the most of, like you said, a little bit less grants, but it seems like you're doing really, it's such a great. There's more to say than just money. It's sort of like the culture.
There's a much more collectivist kind of idea. We're sharing a lot more resources to make it work for everyone. And I think I read somewhere that it might be it's the highest publication rate per dollar that's spent. So
that's great. That's a great metric. I like that.
Yeah.
I should calculate that. Yeah. But so maybe just to ask a few last questions here. So we sort of touched upon, sort of figuring out the brain and trying to figure out what the most, and this is something I grapple with as well, is like what the most meaningful scale is, both temporally and spatially. And of course, the brain is organized across spatial and temporal scales. And obviously the best scale is the highest and fastest you can go, and then you make the most use of what you have. How do you think that things will progress in terms of not only fMRI, but how it's integrated with other methods? What sort of questions are asked? What sort of insights? And then where do you think and how do you think that the first real clinical traction might take place as far as that's concerned? Sort of a blue sky, sort of projecting out into the future.
I mean, that's very challenging.
Yeah. That's an impossible question.
I think that, I mean, I love bold signal. I mean, I think it's a real giver, and it's a scale that I can comprehend.
Yep.
I remember the first time I saw electrophysiology data, and I was just thinking oh my God, this is so much data, it's so fast. Can we just sample this up to every second or two, and so it looks like bold? But no, I mean, there's a whole different set of tools for working with that kind of data, and I think what's going to be really important is how do we integrate this kind of information, and we are going to get faster and faster with our fMRI, and I think it will be very helpful for us to figure out where we can begin to merge the frequencies of information and with electrophysiology.
Yeah.
A lot of the work, we have the capabilities of, simultaneous EEG, fMRI, but I don't think it's really leveraged to its maximum potential. It's kind of we analyze them as separate things. And I don't see a lot of great data integration, multimodal data integration and I think that space will be really awesome.
Yeah, I completely agree with that. We have Pete Malfeas in our group who does simultaneous EEG and fMRI, and he's, right. I mean, it's one thing, just the practicality of doing it simultaneously, but you're right. You're absolutely right. It all comes down to, the best, how do you ask the right questions? Mm-hmm. I think that, for instance, there's this researcher who used to be at the NIH, who's at MIT, Earl Miller, just posts these amazing papers and insights into electrophysiology with humans, and different frequencies, and how that ties into mechanistic models of how the brain functions, and it can tie very much into fMRI. So, yeah, I think there's a bright future for that. I totally agree.
Now, in terms of the clinic, that's a tough one. I mean, I mentioned TMS potentially. I don't know how far we can go with that, but in some ways, I don't think we have to know yet. I think it's okay to stay in the pure research space for a while, and that something will emerge. I think it's a worthwhile continued investment in just understanding brain function, and it will yield its clinical utility in time.
Yeah.
I don't think we have to do that. I don't think we have to solve that yet to justify what it is that we're doing.
No, I totally agree with you. And I think that's a really healthy answer. A lot of people, think that if we press and focus on pressing towards clinical applications, it will somehow happen faster. But I think you're absolutely right. There is a value in just doing research for research's sake and to understand the brain, and it will be clinically applicable when it's ready.
Yeah.
And we'll know. And
But at the same time, the company Serotype, where Prantak Kundu is working and leveraging multi-echo fMRI, it's being used as a potential clinical outcome. So to predict the efficacy of potential medications. So I think that's one potential promise. That it's one more tool for assessing how well things are working.
Yeah, and I think you're right. I think trying to find. I mean, there's a lot of groups trying to find, biomarkers of, just looking, asking the right question. It's not a matter of just collecting tons of data, but it's looking for just the right features, getting some insight and with multi-echo, getting some sort of high fidelity information that you can then, that's actionable. But I think there's not many companies doing that. And I think that will be, all the scanner vendors, they sort of wait and see what's useful and they do it and they explore that space. But it's these third-party companies like Serotype that are trying to, catalyze clinical trials or develop better methods. Or even there's companies that are developing processing platforms that sort of, that help. It's not like the researcher platform where they have work with FSL or AFNI. It's like something that that reduces the degrees of freedom such that there's not variability in how the technologist, adjusts something or whatever. It's just, information. So that's a key part too. But there's so much.
I mean, try getting 10 people who use fMRI to agree on one pipeline.
Yeah. You're right. And that's where people don't appreciate. Like with MRI, you just have, contrast and scale, and that's it. The radiologist tweaks the knobs and he sees it and that's it. But with fMRI, there's so many steps that we don't fully agree upon that are incredibly important. Right. It's tricky. Yeah. A lot of work to be done. All right. Well, I think this was great. This was, I think, one of our record duration podcasts. This was really, really fun, delving into a little bit of depth with your papers, but also just simply talking about these issues, because I think it's important to, sort of take a step back and sort of, think about, I love talking, just kind of riffing on the default mode network but just sort of putting it in perspective and kind of assessing where we are. And I think your work has been really great over the years. And I hope you all, keep on progressing and keep on, doing the right thing. It seems like you've sort of embraced the right combination of questions and methods at the right time. And I look forward to seeing your future work as far as that's concerned.
I appreciate that, Peter. It's been wonderful talking with you. And I'm so grateful for everything that you've provided for the field, from your just basic work with MR physics and pipeline development and of course your stewardship of neuroimage. Like, you've been just an amazing luminary for all of us. So I'm really grateful for the opportunity to talk and share my work, and I really appreciate your interest.
All right. Well, thank you. I'm having fun, and hopefully we'll meet up at meetings in the future and keep on going. So thank you.
Neurosalience is brought to you by the Organization for Human Brain Mapping. This week's episode was produced by Alfie Wine and Farouk Golban.